Energy is the capacity to cause change. And in everyday life, some form of energy can be used to do work or to move against opposing forces like gravity and friction. But it exists in various forms, and cells transform energy from one type to another, maybe from kinetic to thermal, potential energy from chemical energy, but energy is converted from one form to another. As you climb the stairs to the diving board, you're releasing chemical energy stored in cells from the food that you ate. The kinetic energy of muscle movement is converted to potential energy as you climb the ladder. As you dive, the potential energy is converted back to kinetic energy, which is transferred to the water as the diver entered it. And then some heat will be converted, some energy is converted to heat due to friction. Two laws of thermodynamics govern these energy transformations that exist in organisms and all other collections of matter. The first law of thermodynamics states that the energy of the universe is constant. Energy can be transferred and transformed, but it cannot be created or destroyed. This is the conservation of energy. Plants, for example, don't produce energy. They transform light energy to chemical energy. During everyday transformations, some energy is converted to heat, which is the energy associated with the random movements of atoms and molecules. And a system can use heat to do work only when there is a temperature difference. So that loss of energy over time, that loss of efficiency, is part of the second law of thermodynamics. The second law of thermodynamics says that every energy transfer or transformation increases the entropy of the universe. Now, entropy is a measure of disorder or randomness. The more random a collection of matter, the greater its entropy. Although order can increase locally, there is an unstoppable trend towards randomization. In most energy transformations, ordered forms of energy are converted at least partially to heat, and you can use various efficiency calculations, like the one shown in the middle of the screen here, to look at that level of useful output versus not useful output or energy loss. Thousands of reactions occur within the microscopic space of a living cell, and the sum total of an energy's chemical reaction is called metabolism. The set of reactions that make up cell metabolism are complex, they're efficient, but they're also coordinated and highly responsive to subtle changes. So like I said, the energy transformations of life are subject to the laws of thermodynamics. Thermodynamics is the study of energy transformations. In this field, you have the system, which is the matter under study, and the surroundings, which includes the rest of the universe. But when you're looking at these things, we have different components that might make up these situations. So an isolated system, say for example the one at the top here, there's going to be a flow of energy until there is a balance and the energy will stop. In an open system, energy and matter can be transferred between the system and its surroundings. And so organisms are open systems. They absorb energy, be it light or chemical energy in the form of organic molecules, and release heat and metabolic waste products like urea and CO2. Now, when you look at whether these reactions are going to occur spontaneously or not, that enters a new calculation. It's what's called Gibbs free energy. So spontaneous processes can occur without an input of energy, although they need not occur quickly. For example, rusting is a spontaneous reaction, but it's not a fast one. A spontaneous process is one that is energetically favorable. So a process that cannot occur on its own is said to be non-spontaneous. It will only happen if water is added to a system. So how can we determine which reactions occur spontaneously and which ones require an input? That's free energy. Free energy, which is symbolized by the letter G, it's the portion of a system's energy that can perform work when temperature and pressure are uniform. So basically, free energy can be calculated for any specific reaction by applying the equation you can see here in the middle of the screen. So the Gibbs free energy, the delta G, is equal to the delta H, which is the change in the system's enthalpy, which for biological systems is equivalent to total energy. Delta S is the change in the system's entropy or disorder, and T is the absolute temperature. When we say absolute temperature, we mean temperature in kelvins. So if the temperature is provided in Celsius or Fahrenheit, you'll need to convert to kelvins. So for a process to occur spontaneously, the system must give up enthalpy. H must decrease, give up order, T delta S must increase, or both. Basically, delta G has to have a negative value for a reaction to be spontaneous. Every spontaneous process decreases free energy, and the processes that have a positive or zero delta G are never spontaneous. So you can predict what's going to happen if you can calculate that value. We represent that change formulaically here, looking at the change in G. Basically, what you're looking at here is sort of equilibrium. The maximum stability is going to be equilibrium point, and a chemical reaction equilibrium, the rates of forward and backward are equal, and there is no change. At equilibrium, at delta G is equal to 0, a system can do no work. But the more free energy that is available, the less stable there will be, the more work is possible. As things go from a higher free energy state to a lower free energy state, they will be more stable, but less capable of doing work. Taking that further, you can then classify chemical reactions by whether they are exergonic or endergonic. An exergonic reaction, the energy is going to be released, the delta G will be less than 0, therefore the reaction will be spontaneous. If you were to then look at an endergonic reaction, the energy is required, therefore it can be non-spontaneous, and the delta G will be greater than 0. A cell that has reached metabolic equilibrium, that has a delta G of equal to 0, is dead. Okay, so if energy is released during a chemical reaction, then the resulting value will be a negative number, and if energy is absorbed, the resulting value will be a positive number. If you can calculate that, you can determine spontaneity of a reaction. If there are reactions that are exergonic that are going to be giving up energy and reactions that are endergonic that are going to be absorbing energy, efficiency says the best way to combine these things is to couple them. A metabolic pathway begins with a specific molecule that is altered through a series of steps. That is going to then break things down or catabolize them, which is simply an exergonic process where you're going to have energy depleted end products. And then the anabolic processes that are endergonic will take those precursors and put them back together again. By looping these things consistently, you are going to maximize your efficiency. We do this a lot with things like ATP as an energy transfer. So we can see here the fusion of glutamine is going to require ATP to absorb that molecule. The ATP is going to power it by releasing energy. The bond will use some of energy, and the difference, the negative 3.9, will be the energy that is lost due to entropy in this situation. So ATP is a good example of this. ATP is a renewable resource. So we constantly use ATP, but we constantly regenerate it. The free energy to phosphorylate ADP comes from exergonic reactions in the cell, catabolic reactions in the cell. A working muscle, for example, recycles its entire pool of ATP once each minute. So more than 10 million ATP molecules are consumed and regenerated per second per cell. So we get this idea. It's a constant energy transfer, but it's very unstable as it has a high free energy. And as it goes to a lower free energy, as it becomes more stable, that difference can be used for cellular work. If you apply that to an enzyme, what you realize is that enzymes begin with reactants and they turn into products. So in this example, this enzyme is an exergonic reaction. We know that because the beginning has a higher energy state than the end. The delta G is equal to a value less than 0. Overall, though, an enzyme is a molecule that acts as a catalyst, a chemical agent that speeds up the rate of a reaction without being consumed. So in this example, you're going to have a molecule. Energy will be applied to destabilize the bonds. Those bonds will be broken and reformed at a lower energy state, and that is going to be the formation of change. So the energy of activation is a barrier that has to be overcome. This is common in biological systems because even though we have reactions that are exergonic, spontaneity is hard to control. So most biological structures have evolved with this sort of uphill flow, this need for a transition state to give a level of control. When you apply an enzyme, the enzymes have no effect on the reactant energy or the product energy. The delta G is unaffected. The only thing the enzyme does is lower the requirement for the energy of activation. So these spontaneous reactions, these humps for being overcome, help within the breakdown. If you were to look at, say, for example, sucrose, and you were to put it in water, well, we know adding water will break sucrose down into glucose and fructose. But if you wait for it to occur spontaneously, it could take years. But if you add the enzyme sucrase, the natural heat of room temperature is enough to energize that reaction, and the process breaks down within a couple of minutes. So enzymes lower E of A. They make it easier to reach the transition state, but they have no impact on the delta G. If we apply that to the actual structure, what it looks like in an enzyme, basically the molecules, the substrates will enter the active site. That will become the enzyme substrate complex. The catalytic action of the enzyme converts the substrate to the products. Basically, it's a very specific shape match in that active site. And if the active site doesn't fit, then the enzyme will not be able to do its job. But it's going to apply energy to reach that transitional state and reconfigure them into that lower energy state. Okay, so the products will then be released, but the enzymes are catalysts. So it will then be available to be used again and again and again. They are unaffected by the reaction and are reusable. At high concentrations, the active sites on all enzymes are engaged. The enzymes are said to be saturated. So the rate of the reaction is determined by the speed at which the active site can convert substrates to product. The only way to increase productivity at that point would be to add more enzymes. Now, this does get into the idea of, is it lock and key or is it the induced fit model? Hopefully you know lock and key from previous courses, but is it an exact match or is there some inducement of matching? And it turns out there might be some truth to both molecules, but we do know that the binding between the substrate and the enzyme is very, very strong. So it is likely that the induced fit model, which means that the enzyme, once it has a close match, will basically grip and make that match even more solid, will increase that efficiency, is something much more likely to have really occurred within most natural systems. Enzymes function at different rates, though, under different conditions. Basically, evolutionary history has something to say with this. We've talked about this, we talked about denaturation. Remember that enzymes are proteins. So you can denature a protein by applying issues of pH or temperature or salinity. And we can see, for example, that both low and high pH will have an impact on protein formation and structure and denaturation and enzyme activity, but temperature is much more impactful when it gets too high. That applies to a variety of situations. We also find that proteins can be inhibited in a variety of ways. The enzyme has an active site. The active site is where the substrates bind. If the active site is blocked, we call that competitive inhibition. It competes for binding. Now, an enzyme is going to have an equal number of active sites as it does tertiary structures. So a quaternary enzyme with 4 proteins making it up, 4 tertiaries making up the quaternary, would have 4 binding sites, but the more of them that are blocked, the less the enzyme will be able to do. So it's competing for binding. Now there will be other binding sites, and those are called allosteric sites. Those allosteric sites can be bonded to, and when they bond, all of the atoms rearrange, which can change the shape of the active site. That type of interference is called non-competitive inhibition. So we see some of this idea here. There is sort of a selective inhibition, and it doesn't have to be something negative. It could be something positive that the body uses to regulate, or it could be something like a toxin. For example, snake venom acts on the enzyme acetylcholine, the protein acetylcholine and the enzyme acetylcholinesterase. And that means that when the snake venom attacks, it causes a clenching of the muscles, because that's what acetylcholine does, but the acetylcholinesterase enzyme is not able to bind, so your heart will contract and then it won't be able to uncontract again, and that's how the venom can kill you. So there are more advanced forms of regulation that are part of this process, but you can only really see this when you move into the quaternary level. So when you have multiple active sites, you also will have allosteric regulation. This can be a positive thing like an activator, or it could be a negative thing like an inhibitor. Basically, if you remember resonance from chemistry, these protein structures are constantly moving and flexing, and an activator will force the active sites to open. It's still allosteric because it's binding somewhere than the active site, but it stabilizes them, whereas the inhibitor does the opposite. You could also have cooperativity. Basically, when something binds to the active site, it can then stabilize the other active sites, making them more likely to interact. So there's lots of ways that we can affect protein structure and enzymatic activity. This also applies metabolically. So if you look at the larger metabolic change, there can be feedback inhibition. So in this example, we have a biosynthetic pathway. We have threonine, which is going to be a precursor to isoleucine. It goes through a series of enzymatic reactions to produce it. But if you have an excess of isoleucine, it will bind to an allosteric site in non-competitive inhibition and prevent its production. When you start running out of isoleucine, the last ones that you use up will be the ones that are bonded to this enzyme, and then boom, threonine can bind again and more isoleucine can be produced. These are efficiency things. Okay, how did they evolve? Well, the organisms that were the most efficient wasted the least energy and were more likely to survive and reproduce. That's all this here. So it seems like a very controlled process, but it evolved through random accumulation of beneficial traits, which is where natural selection and evolution begins to play its role.